All right, good evening, everybody. I am in my home here tonight, ready to help you review for your cells and cell processes test. I thought I'd rush really fast through maybe 2 screencasts. Each screencast is allowed to be 10 minutes long, so it's about 20 minutes. I don't think that's enough review time. I think you need to review a minimum of 1 hour before every one of my tests. And for those of you that are going for A's and B's, you need to be reviewing like 2 hours. Every time I have a Quizlet or other review materials, you need to go over that. Try those Quizlet flashcards. You need to know every single vocabulary word. It's not that I test you on the vocabulary words, it's that I use the vocabulary words in the test, and you need to understand what the questions are about. And if you don't know the vocabulary, you won't be able to answer the questions. But what I decided to do for these screencasts is to take a look at your study guide and then show you where in the notes we get that information from. I'm going to talk fast. I might not get through everything, but I'm going to give you 20 minutes of review if you're interested. So let's go ahead and start out. So we started out, you took a quiz already on cells in general, cell anatomy, but I just wanted to emphasize a few things. Cell theory, where did we find cell theory? Cell theory was in the notes. Oh, and I'm going to go back and forth between the two sides of the screen. So I have all the notes and the different things that we looked at on the side, on the left side, and then the answers to the study guide on the right side. So cell theory basically says that all living things are made of cells. Cells are the basic unit of life, and cells come from other cells. The puzzling part about that was, where does the first cell come from, KJ? And remember, science doesn't really have an answer to that. We talked about what prokaryotic cells are. Prokaryotic cells are really small. They're bacteria. They don't have a nucleus or organelles, and they are nothing like the cells you are made of, eukaryotic cells, which plants are also made of. Eukaryotic cells are huge. They have a nucleus. They have organelles. All the organelles that you learned in this last unit that are in plant and animal cells, those are part of eukaryotic cells. And then the other thing that we focused on, I'm going to skip ahead a little bit here. The other thing we focused on was the difference between plants and animal cells. You really need to understand that plant cells have things that we don't have. We have less than plant cells have. Plant cells have a cell wall, a cell vacuole, a central vacuole. I call it their swimming pool. And then they have chloroplasts that allow them to do photosynthesis. Remember, chloroplasts are only in the leaves or the green parts of the tree, the parts of the plant that are going to be photosynthesizing. All right, moving on, we'll talk about photosynthesis and cell respiration. Oops, I'm going to scoot ahead. Yeah, I think that's the right set of notes. Nope. Sorry, one more set of notes here. Okay, photosynthesis and cell respiration. The thing that you really need to remember is that photosynthesis and cell respiration are opposite processes. So in cell respiration, that's what's happening in the mitochondria, the mighty mitochondria. Remember, the mighty mitochondria makes your ATP. We take in oxygen. You breathe it into your lungs, your red blood cells carry it to your mitochondria. You eat, and that food is converted into glucose, and it is also brought to the mitochondria. In the mitochondria makes your energy, your ATP, and as a waste product, it produces some carbon dioxide, which you exhale out. And it also produces a little bit of water. Now, take what's at the end of cell respiration and bring that to the front of photosynthesis. Notice the little yellow explosion mark. That's the indicator that I used for energy. Instead of ATP going into chloroplasts, it's light, the energy that goes into chloroplasts. But regardless, light, carbon dioxide, and water go into the chloroplasts of plants, and then out comes oxygen and glucose. And that oxygen and glucose is then cycled over to the mitochondria of a plant. Plants have both mitochondria and chloroplasts. We all are missing chloroplasts. But plants have both. Plants are making the oxygen and glucose for themselves. We're kind of like parasites that eat them and suck up the oxygen that they're making. But they're making the oxygen and photosynthesis for themselves. All right, so then it says from the photosynthesis packet, and I think I have that right here. I thought I had it right there. Shoot, I did have the photosynthesis packet. Here we go. We'll try that again. Let's see if I can get that. There we go. That's what I'm looking for. So it says what enters through the leaves and the stomata. So down here, remember the holes in the leaves at the bottom of the leaf is the stoma. And what comes in is the carbon dioxide that they're sucking up out of the atmosphere. And what escapes out is the oxygen and a little bit of water. They don't want to lose their water. So that's why the stomata have the ability to open and close, is to control how much water escapes in and out. They are located on the bottom of the leaf because the sun hits the top of the leaf and the top of the leaf gets hot. If the holes were located on the top of the leaf, water would evaporate out of the top of the leaf, and that's very bad for the leaf. That's very bad for the plant. So the stomata are located on the shaded part of the leaf so that less water escapes out of the bottom. And then it says, what are the two types of vessels? And this is where you found that. Inside the plant are two different types of tissues. Xylem is one, phloem is the other. Xylem carries water and minerals, and phloem carries the sugars. And then from our CO2 use in plants lab, I think that's this one. That's the one where we blew bubbles in the water and our carbon dioxide mixed with the water and made an acid. When we used that special indicator called bromethymol blue, it turned from a blue color to yellow in that acid, in the carbon dioxide. So it says, let's see, what happened when you blew bubbles into it? It turned yellow because you were adding carbon dioxide. And then why did some of the test tubes change colors? So for example, when we put the plant in it and then put it in the sunlight, the plant photosynthesized, it sucked up the carbon dioxide that you had blown in and it changed the color to blue. Now in this image, there's a green one. That one was probably placed in the dark, and then the plant was not able to suck up the carbon dioxide, so the carbon dioxide stayed in the water. So it says, if you placed a cell that had mitochondria but no chloroplasts into green water, what color would it be the next day? Oh, that's a super good question. It would be yellow. The mitochondria make carbon dioxide. So if I put a fish in the water and left it in the dark or the light, it doesn't matter, the fish would turn the water yellow over time because the fish is producing carbon dioxide. So anything that makes carbon dioxide would cause that water to be yellow. And then the 11th question says, if you placed Elodia into green water in the dark, what color would it be the next day? It would be yellow, and that's one people have a really hard time with. Plants can't photosynthesize in the dark, so they're not going to suck up that CO2. And hang on to this because this is really hard to remember, they have mitochondria also. So in the dark, they're actually making CO2. So if you leave that plant in the dark for long enough, the water will be yellow. All right, next, notes from osmosis, diffusion, and active transport. So that's this set of notes right here as it comes up. There we go, the diffusion, osmosis, and the following ones. So the first thing it says is define homeostasis. Remember, the prefix homeo means same, and the suffix stasis means same. So it's maintaining this stable internal environment or balance within the organism. Examples of maintaining homeostasis are things like water, making sure that your amounts of water are even so that you pee and you drink so that water always stays the same, or your body temperature is sweating or shivering to stay warm, or sweating to stay cool, are causing your body to maintain that constant 98.6 temperature. All right, number 15 says write active or passive by each example, sorry. And that's both a combination of both of these sets of notes. So we had the active versus passive cell transport. And the thing to really remember is that active transport uses ATP. So here's that picture. It's a cell membrane. We have some molecules going across without the use of a protein. That's called passive transport. We have some molecules going across using a protein. That's called facilitated diffusion. It's still a type of passive transport because no ATP is being used. But in this case, some molecules are going the wrong direction. Notice they're going from a low concentration to a high. That's the opposite direction. That's active transport because ATP had to be used. So let's take a look. Uses ATP, active. Osmosis and diffusion, those are both passive. The movement of CO2 across the cell membrane, that's passive because CO2 is small. I just got a warning that my time is up for this screencast. I'll finish on screencast number 2. See you in a moment.